again, Universal Science. Welcome back to microbiology. Today we'll be discussing the theory of spontaneous generation, and here are the theories of various scientists that contributed to the theory of spontaneous generation. Okay, so basically, we're going to start by defining the theory of spontaneous generation, which is also known as the theory of abiogenesis. What does the theory of spontaneous generation say? Well, basically, it's trying to tell us that living things, as in life, as in me, myself, I came from a non-living thing. Do you understand? Like, they are trying to tell us that wood now can give rise to me, a living thing. That's every living thing. In definitive terms, you say the theory of spontaneous generation is trying to say that, or rather, it says that every non-living thing have a life force in them, meaning that every non-living thing you see all around you, wood, chairs, they have the ability, they have the force in them to give rise to a living thing. And various scientists, such as we have the Pasteur experiments, we have the Leeuwenhoek, we have Lazarus Polanzani, and here we have Pasteur, which is regarded as the father of spontaneous generation, because it was his own theory that finally disproved the theory of abiogenesis, which is spontaneous generation. Alright, let's dive into their specific theory and explain why it was and wasn't validated. Performed an experiment on the theory of spontaneous generation. What was his aim? You know, when you are doing theories in biology, in science, you use this unit called A squared mod. You make use of this unit called A squared mod, where A represents the aim of the theory. Other A represents the apparatus used in the theory, which you can see here on the board. Apparatus used. Then M represents method, which was the method applied in performing the theory. Method. O is simply the observation of the experiment. And C is the conclusion. That's the conclusion of the experiment. C is conclusion. Alright, so let's begin by discussing the theory of spontaneous generation based on Francisco Redi's study. What was his aim? His aim was to validate the theory of spontaneous generation. What was the apparatus used? He used beakers. He used 6 beakers, but out of the 6 beakers, 2 of each were repeating the same experiment. The remaining 3 beakers, which are repeating this same exact experiment, are referred to as the control method, or the control theory, the control experiment. You know what that means, right? He wants to perform the experiment twice to make sure that everything he's going to document validates it. So now he made use of 6 beakers. He made use of a Koch stopper. He made use of a broth. You know what broth is? We know what broth means. We watch movies. You know, broth are like salad, soup. You know, broth are soups. Basically, broth are soups. Then he made use of a gauge or a mesh. So now what did he do? What was his method? I've said his aim. I've said his apparatus. What was his method? His method. He brought in broth and filled these 6 beakers. But I'll be saying 3. Now, but we all know that it was 6 beakers that was used, but based on what's on the board is 3 beakers, I'll be using 3. He filled these beakers with broth, then he left one open, he covered one with a Koch stopper, and he covered one with a gauge, or you can call it a mesh. And then, later, he observed that on the one he left open, he found maggots on top of the broth, like maggot was inside this broth. Then on this one, he covered with a Koch stopper. He found out that there was no maggot at all. Then on this one, he covered with a gauge or a mesh. He found maggots on top this gauge and on top the mesh. Okay, so now that was his own conclusion. Then he concluded that the theory of spontaneous generation is false. Why? Because maggots cannot grow on this one. That means he was trying to tell us that the reason maggots grew on this and this is as a result of miasma, which we call air pollution. That miasma was the reason maggots was able to grow on this this experiment, and same reason why maggot was able to grow on this experiment. But this one was properly covered, and that was why miasma could prevent the growth of microorganisms. And then his theory was not validated because another scientist came, another scientist named John Needham, and then he repeated this experiment again. He repeated the same experiment again. Then when he repeated this experiment, this time around, he boiled the broth, he did the same thing, did the same exact experiment. Then what did he find out? His aim was the same thing, to validate the theory of spontaneous generation. The apparatus made use of a Koch stopper, a broth, a beaker. Then what was his method? He boiled the broth, put it inside here, and he covered it with a gauge. Then he found out that his observations were maggots grew inside this broth, and his conclusion was he supported the theory of spontaneous generation. But why was his theory wrong? His theory, scientists came up and they were like, no, this theory can't be right because he probably did not boil the broth well, and that was why microbes were able to grow inside this experiment. Then that scientist came out and said, okay, since that's the case, I want to perform another experiment, and this time around, I'm going to be validating or disproving or supporting the theory of spontaneous generation. And then this experiment, his name is Lazarus Spallanzani. Oh my god, that name is so cute. Lazarus Spallanzani. Okay, so Lazarus Spallanzani came and said he wants to repeat. He repeated this John Needham's experiment. This time around, he sterilized the beaker, he sterilized everything. He boiled the broth well, covered it. That was his aim, was to validate the theory of. When I mean validate, I don't mean support though. Validating could be like disproving the theory, it could be supporting the theory. So his apparatus, he made use of beaker, he made use of a Koch stopper, a broth. His method, he boiled and sterilized all these apparatus very well. Then his observation, he observed that broth and maggots did not grow inside this broth. Then what was his conclusion? His conclusion supported his experiment was against the theory of spontaneous generation. What was his experiment against the theory of spontaneous generation? Well, many scientists came up later and they were like, he overboiled the broth and made the whole experiment airtight. And of course, for we to continue living, for life to exist, we need oxygen. So that was why his theory was not validated or supported as the major theory to disprove the theory of spontaneous generation. Then lastly, we have this wise man. His name is Louis Pasteur. Of course, his name is Louis Pasteur. And then this man came, he sat down. Oh my god, oh my days. He sat down and he thought, what can I do to make this experiment? What can I do to actually make a significant impact on this same experiment that various scientists, when I mean scientists, scholars have been trying to prove? Then he came and he brought up this swan neck experiment. Swan neck experiment. They can ask you, what experiment or what method did Louis Pasteur use to disprove the theory of spontaneous generation? That's the swan neck experiment. Then this experiment, this is the setup of this experiment, where here he made this space, air could not enter through this. Then here, when he filled this place with broth, after boiling the broth, putting it in this, he filled this place, then he was boiling here. Of course, air was able to enter through. That was the main reason of this thing, for air to be able, for oxygen to be able to enter. So even if air enters and then miasma or maybe microbes or anything enter with it, they are going to stop here. Because this fire here is to kill microbes and they will not be able to have the ability to swim up to this place. So his experiment, after performing his experiment, he observed that microorganisms did not grow in this broth. So what was his conclusion? His conclusion disproved the theory of microbiology. And there was one other last scientist named Pierre Michel. She's not very popular because her experiment did not really prove major things in this. Our experiment was only trying to explain the spores of fungi. Well, let me summarize the whole sciences and whether their theory was with or against theory of spontaneous generation. So like we said, we have Francisco Redi. Remember in the experiment, like I said, this one's experiment was against the theory of spontaneous generation due to the inability of microorganisms to grow in the experiment covered with a Koch stopper. So the theory was against the theory of spontaneous generation, which I'm representing as SG over there. Then we also have John Needham. Their names are different, right? John Needham. His theory was supported the theory of spontaneous generation, supported SG. And we all know the reason his theory was not accepted because he did not boil the broth properly to be able to kill all microorganisms within that broth, because a microorganism possesses endospores that can make them resist heat. Do you understand? We have some microorganisms that are thermophiles, like the thermophiles, they can resist heat, or we have the mesophile, they can resist moderate temperature. So you have to boil it in such a way that you will boil it. Then we have Lazarus Calandani. I don't know that name. Lazarus Calandani. Lazarus Calandani's experiment was against theory of spontaneous generation. And why this theory was not validated was because he probably overboiled the broth, and of course, for life to exist, we need oxygen. Of course, we need oxygen. Then last, this able president named Louis Pasteur was the person, the scientist who came and disproved the theory of spontaneous generation. Of course, that man needs accolade, because how will you tell me that I came from spoon? How will you tell me I came from wood? So please take note of this, because they might ask you your OBJ question that which of these scientists disprove the theory of spontaneous generation. Disproving the theory of spontaneous generation is different from which of these scientists were against the theory of spontaneous generation. So the scientist that disproved the theory of spontaneous generation is Louis Pasteur, which is now regarded to as the father of abiogenesis or the father of spontaneous generation. Why these scientists here? They supported and they were against the theory of spontaneous generation. So Louis Pasteur here disproved the theory of spontaneous generation. So here we are done with theory of spontaneous generation, and we're done with conflict of spontaneous generation, which are these experiments which various scientists came to perform. They deserve an accolade. Lest I forget, there's also another scientist. Her name is Pierre Micheli. Well, her experiments did no significant good to the theory of spontaneous generation, but of course we need to give her accolades because she did something. Her experiment was only trying to explain the spores of fungi to us and all that. So I came was talking about spores. So now we're done with theory of spontaneous generation, and we're done with conflict over spontaneous generation. Thank you for staying. Please kindly subscribe to our YouTube channel. Meet you in the next video.